Okay, thanks for joining us today for this professional development webinar on what Flatiron is doing to harness the power of data science and real-world evidence for cancer treatment, access, and care. My name is Karis, and I'm part of the Public Health Alumni Association Board of Directors, and this is a webinar that we put on for all of you. Today's agenda, I'm going to tell you a little bit about the Public Health Alumni Association Board of Directors and some of our upcoming events. Then I'm going to tell you about our amazing speakers we have today, and we'll jump into the webinar. So, the Public Health Alumni Association Board of Directors is from UC Berkeley School of Public Health. It's been around for a long time, since 1953, and our goal is to really build relationships across alumni, students, and others in the public health community. And we have a volunteer board of directors. You can find our website if you like. We have a bunch of different committees. There's one on scholarship, diversity and inclusion, events and networking, and this webinar is from the professional development subcommittee. And you're welcome to go see more about that. We have a couple other webinars that'll be coming up a little bit later, probably early in 2019. We'll have one in the general field of global change in health and one on inequality in health. So stay tuned for those. We also have a couple networking events that are coming up in spring. 2019, we'll be doing a joint event with the Women Healthcare Executives Organization and PHAA, probably in February or March. And we'll also be doing another joint happy hour, as we've done before, with several schools of public health in May. If you are not already getting notifications about events like these and you would like to be notified, you can email me. My name is Karis, and my email address is there, ctakaro, CTAKARO, @berkeley.edu. And we would be glad to have you join us. So I want to tell you about our speakers today. We have two really exciting speakers. One is Meghna Samant, and she oversees a group of data scientists who are working on real-world observational studies and clinical genomics data applications. So before coming to Flatiron, she worked at Roche Genentech, where she managed statisticians working on oncology clinical trials, and she also held cross-functional leadership positions in clinical development. Meghna is a biostatistician by training and received her PhD in biostats from UCLA. We also have on our presentation team today, Raina Mathur, who works on real-world observational studies in oncology with a regulatory impact. And she's got a wide variety of experience in drug development, including lab-based research in preclinical studies, management consulting in clinical trials operations, and health outcomes research in both infectious and chronic diseases. And she is a Cal Bear and received her MPH in genetic and molecular epi from Emory University. So you're going to love these two speakers, and I just want to talk a little bit about logistics before I hand it over to them. We will have time set aside for Q&A at the end of the presentation, and the way you can enter your questions is through the chat box. You should be able to see a chat box probably at the bottom of your screen that looks like a little speech bubble. Go ahead and type in those questions at any time, and I will keep track of them, and I'll read them out for our presenters to answer at the end. You are all muted by default, so if you want to go ahead and eat your lunch, no one will be hearing you. We are recording this webinar, and if you would like to be notified when we have the link and the slides posted, again, you can contact me, Karis, ctakaro, @berkeley.edu. And I know that Zoom says that I'm Eileen Pearl. That's because Eileen set up our Zoom account for the Public Health Alumni Association, but I'm actually Karis. So with that, I would like to turn it over to our speakers.
Hi, everyone. I'm going to take a second to share my screen really quickly. Okay, can everyone see this?
It looks good, Raina.
Okay, thank you so much.
Okay, hi everybody, Meghna Samant here. First of all, I want to take the opportunity to thank Karis and the UC Berkeley School of Public Health for giving us the opportunity to come and talk to you about real-world data for cancer treatment, access, and care, and particularly our work at Flatiron, how that fits in, and opportunities to contribute to the greater research. So just a brief outline of our presentation. We'll introduce introduce real-world data and also make an introduction to Flatiron as a company. Then we'll go into quantitative sciences, who we are, what we do, and this is really our home department. And this is our home team where we sit at in Flatiron. Raina will talk about just her personal experience on working at Flatiron, and also the opportunities for those interested potentially in joining Flatiron. So with that, I'm going to turn it over to Raina to introduce real-world data and go from there.
Cool, yeah, thank you. So we wanted to start off our talk today by describing how patient health records have evolved over time. So in order to do this, I want everyone to think about when they were little and they went to the doctor's office. And imagine that you're sitting there in the examination room, and in walks the physician or the nurse, and in their hands, they're holding a big fat manila folder with your name on it. And inside, it's stuffed full of papers of every size and shape and color. And when they sit down and ask you, where does it hurt, or what are your symptoms, and you give them an answer, they're taking pen to paper and writing all this information down. This was the status of paper health records in the past. So everything was recorded by hand on paper. Naturally, this is a very low tech, low cost, and low maintenance solution to collecting patient health data. But you can imagine that it's cumbersome to store and transfer information in this way, and much harder to retrieve. From a data analysis perspective, it's also a lot harder to aggregate information across cohorts of patients with specific clinical characteristics and drive data insights in this way. This really all started to change with the passing of the 2009 High Tech Act. And for those of you that are unfamiliar with this particular piece of legislation, it was enacted as part of the American Recovery and Reinvestment Act that was signed during the Obama administration. And the purpose of this piece of legislation was really to drive uptake and use of electronic health records, or EHRs. So this is what's driven us to our current state. Currently, when you go to the doctor's office, they might walk in with a laptop, or there might be a computer set up in the examination room. And now, when you describe your symptoms to them, they're manually typing it into an electronic health record. This piece of legislation drove adoption of EHRs in oncology specifically from 10% adoption to 95%, which is incredible. EHRs are naturally higher tech, higher costs, and require higher technical maintenance, but you can imagine that it's much more efficient to store patient health data in this way. And again, from a data analysis perspective, it's much better suited to drive data aggregation and data insights. So I want to pause here and describe the tie between electronic health records and real-world data. Real-world data here at Flatiron is exactly this. It's patient-level health or clinical information that's collected from an EHR. And really, the cadence that we're able to collect this information is dictated by how often the patient goes in to see their physician. And the level of completeness is driven by how well the physician is documenting that patient's health status. And I'll contrast this with data that comes from clinical trials. Data that comes from clinical trials is collected on a very strict follow-up schedule, and any clinical variable or data element of interest is captured to pretty much near 100% completeness every single time that patient is seen. So one of the challenges that we face here at Flatiron is to demonstrate that you can use real-world data to drive the same insights and analyses that you can using clinical trial data. So more formally, real-world data is data relating to patient health status and or the delivery of healthcare routinely collected from a variety of sources. You've already heard me talk about the fact that here at Flatiron, our real-world data comes from EHRs, but you can also collect this type of data from insurance claims or even federal databases like the Social Security Index or the National Death Index. Real-world evidence is evidence derived from real-world data through the application of research methods. Or to think about this more simply, it's basically the results from an analysis that is run upon real-world data. And some of the research questions you can ask in our Flatiron data is, what are the outcomes of patients with a particular disease and specific clinical characteristics? How well are specific drugs working in specific cohorts of patients? Can we look at safety events or adverse effects associated with specific drugs? And how can I evaluate compliance to specific drugs? And here at Flatiron, we have both a hand in creating and curating the real-world data. And even from the quantitative sciences team's perspective, we have a large hand in translating real-world data into real-world evidence. So what is Flatiron? Flatiron is a healthcare technology company devoted to generation, aggregation, and analysis of real-world data to drive powerful insights to improve lives of cancer patients. As such, we are a 100% oncology-focused company, and we currently are about 650 full-time employees, but are definitely growing. We are cross-functional as a company, so we comprise of software engineers, data scientists, oncologists, product managers, really people from a vast variety of backgrounds. We're headquartered here in New York City, but do have a presence on the West Coast in San Francisco. And we were acquired by Roche in February of 2018. And Flatiron Health's overall mission is to improve lives by learning from the experience of every cancer patient. One of the ways in which we do this is we build software for providers. One of our products is called OncoEMR, and it's a cloud-based oncology-specific electronic health record system. And we market this towards community oncologists who can adopt this into their practice and into their workflows. The second product is called Flatiron for Academics, and it's an analytics platform for academic institutions who maybe can't adopt OncoEMR due to strict rules at the institution, but they still can run analyses using our real-world data and contribute to our real-world data. And what you're seeing here on the slide is a map of where both of these products have been adopted across the US. And as you can see, it's pretty widespread. This next slide summarizes the implication of that widespread adoption across the US. So as a result, in our real-world data sets, we have access to 2.1 million active patients that come from 2,500 and more clinicians, 280 cancer clinics, and 800 unique sites of care. One of the unique things about Flatiron data is not only that it's composed of millions of electronic health records in a single common data set, but it is composed of both structured and unstructured data. Structured data is data points or data elements that are organized in a predefined manner. So one such example of this is when a physician goes in to record a patient's birthday. What they do is they go into the EHR and there's a specific designated field for them to input the birth date of that patient. And not only that, but it forces the physician to adhere to a pre-specified format. So a 2-digit month, a 2-digit day, 4-digit year. Unstructured data is any information that's not organized according to a pre-existing data model or pre-existing format. So an example of this is any information that's collected in free text form from a pathology note or a physician note or a lab report, etc. And we want to go a little bit into more detail about why unstructured data is important. And so in order to do so, I'll give you guys a hypothetical scenario. So let's say you have a physician who has a new patient, and this patient was recently diagnosed with some form of advanced cancer. So the physician knows that they need to start this patient upon a therapy right away. However, this physician is a little bit unclear which therapy would be best suited for this patient. Fortunately, in the literature, it says that depending on the patient's biomarker status, in this case, that biomarker is PD-L1. So depending on the patient's PD-L1 biomarker status, there may be a therapy that is more efficacious for them. So the physician says, okay, I need to take a biopsy or a sample of this patient's tumor and figure out what their biomarker status is. And the results of that biopsy and that test are what we're seeing on the screen right now. So this is a pathology report. And from this pathology report, we can see that the physician went into the lungs, specifically the right upper lobe, and that the patient's PD-L1 biomarker status is negative. So using this information, the physician is able to make a treatment decision for that patient. Now, the key thing here is that the pathology report is only attached to the EHR. This information is not specifically entered into a pre-designated field. So if we do not look at the unstructured documents and find this unstructured data, we would never know this piece of critical information about this patient that drove their treatment decision. Furthermore, you can imagine that there are individuals on the research side who would be interested in investigating the comparative efficacy of a drug in cohorts of patients that are biomarker status positive versus biomarker status negative. So that's why here at Flatiron, we are really passionate about collecting this unstructured data and being able to provide that to our clients and to our researchers. The way we do this is through technology-enabled abstraction. So at Flatiron, we have a team of expert abstractors, and I think to date, the size of the abstraction team is like over 1,000 people. It's really quite huge, and everyone is trained in oncology in some way. So these individuals were formerly oncology nurses or certified tumor registrars or any other oncology clinical research professional. And they go in and they manually review all of these unstructured documents that are just an attachment to each patient's electronic health record. Flatiron technology is really this layer on top of that that facilitates the abstraction process. And some of the things it helps them do is performing things like document classification or visual organization, and also enabling text search within documents. And it's all to just improve the efficiency of that abstraction process. So we've applied this format of combining structured and unstructured data to multiple malignancies. Back in 2014, we had only 2 longitudinal real-world data sets, one for advanced melanoma, a type of skin cancer, and one for advanced non-small cell lung cancer. And as time has gone on and we look to 2018, we've increased that number to 18 longitudinal real-world data sets. And as we look forward to 2019, that number is only going to grow. Another one of our cool products here at Flatiron is the Clinical Genomic Database, or CGDB. And this was really driven forward by a partnership between Flatiron Health and Foundation Medicine. Foundation Medicine is a company that has information on comprehensive genomic profiling across hundreds of cancer-related genes for each patient's tumor. And what we've done is couple this information with our real-world longitudinal patient-level clinical data from the EHRs to form the most comprehensive data set to encompass the patient's disease course to date. And the CGDB currently encompasses 40,000 patients or so, but is very quickly growing. So with that, I will hand it back to Meghna, who will describe the quantitative sciences team, who we are, what we do, and also give you some case studies about our work.
Okay, thanks, Raina. So I'm going to talk about our team of quantitative sciences. And essentially, we are a group of about 30 scientists that impact patient lives through our deep understanding and translation of real-world data to generate evidence. We really considered the analytical and methodological hub of Flatiron, and we come from diverse backgrounds. I'm a biostatistician by training. Raina is an epidemiologist, and we have several colleagues who are health economists, computer scientists, computational biologists, or have other advanced scientific and quantitative trainings. Below you can see some of the institutions from which our staff comes from, and Berkeley being certainly one of the prominent ones. So what do we do? Our work can be divided into really three key buckets. First, we transform real-world data into high-quality functional data sets for cancer research. This is really the meat of what we do at Flatiron. But we don't stop there. We design and implement robust statistical analysis plans and apply quantitative methods to analyze and visualize that data. And third, we provide quantitative leadership in cutting-edge research related to real-world data, working in collaboration with other Flatiron team members. So we typically work in highly cross-functional environments. So on any team you take at Flatiron, you'll have quantitative science sitting on it, in addition to an oncologist, a software engineer, somebody from product, from operations, etc. Some examples of the kinds of things that we are doing, and I think it's important to note here that the real world data, and as Raina mentioned with the High-Tech Act of 2009, is relatively recent, and analysis and understanding the analytical methods for appropriately analyzing this type of data is absolutely critical. So some examples of more methods type of work that we do are given here. First, we evaluate bias using statistical methodologies. So, thinking about, so those of you who are familiar with propensity score weighting or inverse probability of treatment weighting, informative censoring and the bias that can bring in survival analysis, thinking deeply about cohort selection and survivorship bias, etc. So when we are dealing with these data sets, we do a lot of deep digging and thinking about all the kinds of biases that can impact the analyses. Another example is we assess feasibility and performance of real-world control arms to supplement or complement clinical trials. So let me explain a little bit what that means. So many of you may be, familiar with randomized clinical trials, right? Where you have an experimental regimen, which is compared, the effectiveness or the efficacy and safety of an experimental regimen is compared to that of a control regimen. Many a times, having two arms in the trial can make the trial expensive or even lengthy. So one strategy that many pharma companies have undertaken is to run single-arm trials where they test their experimental regimens and then use real-world data or other sources to serve as the control arm, to serve as the benchmark for testing the experimental regimen against. But understanding whether the real-world data can be used as an effective control arm needs a lot of work, especially you can imagine making sure that the patients are similar, we are collecting outcomes in a similar way, etc. So we do all that assessment. And third, part of that is also defining and validating real-world endpoints. Mortality or death data being one of them, real-world progression and real-world tumor response. Now so death, progression, and tumor response are common endpoints in oncology. And so we need to be able to understand what does a real-world progression mean when it is being compared to a progression that is coming from a clinical trial, which is typically subject to a rigorous criteria and a standardized criteria. So as you know, like in the real world setting, there is no standardized criteria that oncologists use to determine whether a tumor is growing or not. They look at, radiographs or scans and make their best judgment on, is the tumor increasing in size or decreasing? But they don't go into the granular details of how much it is decreasing, etc., which is a criteria like RESIST, for those of you who are familiar with oncology research, really dictates at a much more granular level. One endpoint that many of us think might be very straightforward and complete and clean in the real world data is mortality, right? After all, death is a binary variable. Either you're dead or not. And it should be, collecting the date of death should be the most, should be an easy thing, right? A straightforward thing to do. But actually, it turns out it's not. So I want to maybe focus on that a little bit, give you an example of the kind of work we do. So we have had to undertake an entire validation of the real world mortality endpoint. And let me explain what that means. So for real world data, it turns out that a complete and timely source of death data does not exist. But as you can imagine, it's critical for cancer research, right? Fundamentally, we want to know, are our patients living longer, because of certain treatments or any kind of interventions? There are two primary sources of death data in the United States. SSDI, or the Social Security Death Index. Till 2011, all states in the US were mandated to report that information. And since that mandate was taken away in 2011, we have seen a decrease in the reporting of deaths. So you can just see, and you can imagine the amount of missing data then that this is now subject to. The second one is called the National Death Index. This is actually considered the gold standard for mortality data, but this suffers from recency. So typically you see a lag of anywhere between 16 to 18 months. And as you can imagine, it's not ideal by any means to have data which is a year and a half old to conduct some contemporary research. So at Flatiron, we thought about how do we create a variable that we can analyze which has highest completeness as well as is timely and timeliness for death information. So our approach is to develop a composite variable while maintaining the versioning. And I'm giving you this as an example because quantitative sciences has been at the cutting edge of this. This is members of our team have led the development of this variable. So here what we see is we started with version 1.0, where we took the date of death that was reported in the structured fields, and we also linked it with a commercial data set that we bought. We quickly realized that the date of death is actually not routinely collected. So and in oncology, if you had an oncology clinical practice, once a patient moves from the oncologist's care to hospice and then dies, typically that information doesn't get back to the treating oncologist, and the oncologist does not enter. Many a times that information does not get entered in the electronic health records. So that creates a lot of missing data. Having said that, there might be times when the families of the patients might send a thank you note to the doctor saying, such so-and-so passed away on such and such date, and thank you for your care. And that note might get scanned, and that becomes part of the patient's electronic health record system, right? So our version 2.0 took that aspect into account. And so we added the abstracted death information from unstructured documents like a thank you note or an email or whatever. And we also linked the SSDI, the Social Security Death Index. We have a publication out that was spearheaded by a member of our quantitative sciences team. If you're interested, Curtis et al. In Health Sciences Research. And then, but we are not satisfied. Where we are heading is now taking the 2.0 variable and then actually improving the way we collect date of death in the EHR. So that'll be the 3.0. That's where we are headed. In terms of, like I've talked about creating these versions, like version 1.0 variable, version 2.0, and so on. In terms of understanding, how good these are, we measured the quality using 3 key metrics: sensitivity, specificity, and accuracy, using the National Death Index data as a benchmark. So basically, sensitivity measures assess, what % of patients known to have died in the National Death Index have a death date in the Flatiron database, right? So they're also known to have died in the Flatiron database. And that's really important. That really talks about the completeness of our death data. Specificity says patients who are not dead, how many of those are, do not show up as having died in the Flatiron database. And accuracy is, do we agree on the death date? So in terms of sensitivity, you can see these histograms here. The gray bar is the sensitivity that you get when you use the Social Security Death Index. And it's not surprising that the sensitivity is so low, because if you remember, I showed you the graph of declining, reporting of deaths. When we use the version 1.0 variable, it moved to about 80%. With 2.0, it has moved to 90%. And we are hoping that with future versions like 3 and 4 and so on, the goal is really to get to that 100% so that we can have as complete information as possible. We've done, I think all measures have done pretty well on specificity and accuracy, so we won't go into a lot of detail there. So that was just one example of the kind of leadership that quantitative sciences has taken, right? Now let's switch gears a little bit about, speaking about our data, how are others using this Flatiron data? What are the broader use cases? And actually, really, the use cases span, all areas from, research, commercial, regulatory, lots of applications. So, for example, pharma companies may be, interested in understanding uptake of new therapies in the real world setting, in the overall population or in specific populations. Or we may want to determine the correlation between treatment sequencing and survival. A basic research question could be, are patients living longer if they receive drug A and then drug B versus if they receive drug B and then drug A, right? And these are the kinds of questions you can ask using Flatiron data. And then one of the more exciting parts is the regulatory application, where submission of real-world outcomes can be done to support label expansions and also support reimbursement. So I want to give you an example where one of our main customers, Roche, used Flatiron data to secure reimbursement for one of their lung cancer drugs. It's called alectinib. Using real-world data. So what happened was Roche ran a single-arm study, clinical trial of alectinib, and in order to secure reimbursement in Europe, needed to demonstrate superiority of alectinib versus the standard of care ceritinib. So using Flatiron data, Roche generated an external control arm of patients receiving ceritinib and compared that to the alectinib data. Since the patients could have, there could be important differences in the patient populations between the clinical trial and the Flatiron data set used, Roche used, IPTW, or inverse probability of treatment weights, from a propensity score model to adjust for those differences. The results were presented at the 2017 European Lung Conference, and the results helped to expand access in 20 plus European countries. And I'll show you in a minute what those results were. But the most interesting part of this story was that while these results were being generated and presented, the results of the ASCEND-2 trial read out, which included an arm of suritinib alone. And Roche did an analysis comparing the results of the suritinib arm from the ASCEND-2 clinical trials with the real-world data, the suritinib arm using the Flatiron database. So this is the poster that was presented, and here are the results. What you're seeing here are Kaplan-Meier curves, for those of you who may be familiar with survival analysis, where the x-axis is time and y-axis is survival probability, right? So the idea is the higher the curve, the longer the survival probability, longer the survival, and the more effective the treatment regimen. The gray line here, or the gray curve, is the alectinib arm. The green curve is the suritinib arm that was created using Flatiron data. As you can see, and what you see here is the median survival is about 15.6 months using suritinib, and 26 months using alectinib. So these results showed that alectinib was superior to suritinib. Then, when you superimpose the suritinib arm from the ASCEND-2 clinical trial, what you see here is remarkable. In particular, the blue curve now, which is using the ASCEND-2 suritinib arm, is virtually indistinguishable from the suritinib curve using the Flatiron data. So that shows, and I mean, this was a really great example where you could really show that an external control arm could be generated using real world data, and in particular Flatiron data. For those of you who are into genetics and deal with genomic data, a very quick example of a research question that we asked internally within Flatiron. And really, our research question was to identify patterns of resistance to EGFR inhibitors in lung cancer. EGFR, or epidermal growth factor receptor, is an important driver in the growth of lung cancer. And effective treatments inhibit that particular biomarker. So they're called EGFR inhibitors. So while these EGFR inhibitors are quite successful at controlling progression and prolonging survival, many patients develop resistance. And so our research question of interest was to identify the patterns of resistance. And in particular, we wanted to understand, are there other biomarkers that get upregulated or downregulated, that might provide resistance to these treatments. So to do that, we used the Flatiron Foundation Medicine database to identify post-treatment changes in genomic profiles of tumors. So we had tumor biopsies, and we looked at the genomic profile pre-treatment and compared to the genomic profile post-treatment, and we identified what those changes were. The interesting thing was that what we found was that the results were largely consistent with known mechanisms of resistance that have been published in the scientific literature. And in particular, one key resistance mechanism that was shown by the data was a mutation in the T790M gene, which is a known resistance factor, for EGFR inhibitors, and that was based on clinical trial data, and then we were able to replicate that finding using real-world data. This was a very interesting piece of work that was recently presented at the American Association of Cancer Research conference in earlier this year. This is the poster that we presented. So I hope through these examples, we have given you a flair of, what that how Flatiron data can be used, and some of the analysis techniques and leadership that quantitative sciences, is assuming at Flatiron to move the field forward. So with that, I'm going to turn it to Raina, who will talk about a personal reflection about working at Flatiron.
Yeah, so after hearing about the case studies of the work that we do within the quantitative sciences team at Flatiron, I thought it might be useful to kind of bring that back to what that means on a day-to-day for a quantitative sciences member, and also speak a little bit about the culture here overall at Flatiron. So I think we've said this a number of times during this presentation, but here on the quantitative sciences team, we really do work cross-functionally. So we are in constant collaboration with our research oncologists, our software engineers, our people in the life sciences partnerships team who are responsible for interfacing with our clients, and also working very closely with our clinical data analysts, who really are the experts in the individual patient level records. And on a day-to-day, we're working constantly to define data models, which entails ensuring that variables are clearly defined and that they're analytically useful, both to our data scientists here at Flatiron and also to the data scientists over at our clients. We run pilot studies to ensure that these variables can be reliably abstracted from the patient record, and that when we do that, they actually measure what they're intended to measure or capture. And we also compare these variables that we derived from the patient record to external data sources to ensure validity, which speaks exactly back to what Meghna presented about the validation of the mortality variable. We also do that for real world progression and for real world tumor response. Furthermore, we are responsible for developing analytic guidance for our internal users and for our clients. So advising people on what is the best way to use our data and how in your analyses can you minimize biases, and knowing that this data comes from a real world data source, what are some limitations of our variables that need to be kept in mind? And then lastly, but not least, we do design and carry out our own analyses here in the quantitative sciences team. And if I think a little bit more broadly about this and speak to the culture here at Flatiron, I personally had, 3 main career goals for myself coming out of my master's program. The first one was that I wanted to continue to learn and apply complex statistical methods that are commonly applied in outcomes research. I wanted to have opportunities to develop presentation skills, communicating analyses to both internal and external audiences. And through all of that, I wanted the opportunity to have ownership over these initiatives. And I can honestly say that the culture here at Flatiron really speaks to all 3 of these things. And it does that by, number 1, providing unmatched mentorship. I think when I entered this team on my very first day, I was given a person assigned to me that helped me learn anything that I needed to learn. That person was there for me anytime I had a question, even if it was the stupidest thing I could have asked. And another thing here that I think is really key is that we operate in what I've called a hierarchical-less fashion. So that means someone who's more junior in their career, such as myself, can really lead analyses or client-facing projects without fear. I can go to someone and say, hey, I would be really interested in leading that work. And there's someone here who will be my advocate for that at all times. And the third thing that I really appreciate about the culture here at Flatiron is that we are constantly pushing each other to be creative in devising solutions to methodological problems. So speaking directly to some of our endpoint validation work, if I'm putting together an analysis plan to validate the variable, there's someone who's going to be next to me saying, hey, have you thought about this method or this method or this method? Constantly questioning the work that we're putting forward and just constantly iterating to make it more valid. So with that, we wanted to transition into describing the opportunities that we have here at Flatiron. So Meghna and I, you've heard us say over and over again, we are here from the quantitative sciences team, and we are hiring both for our San Francisco and our New York City offices. Provided a link to our job postings here, and I think that those slides will be distributed afterwards. So if you're interested, we really hope that you will go check out our career website and apply. If you think that maybe your skill set currently is not maybe suited the best towards quantitative sciences, but you would want to investigate other open positions in our other teams at the company, please again do visit our career website, and a link again is provided here. If you want to reach out to Meghna or I directly, we've provided our email addresses here. If you have any questions about the work or would even like to provide us with feedback on this presentation, we'd love to get that from you. Or even look us up on LinkedIn. We'd love to hear from you in any format. So yeah, thank you so much for being here today, guys. And I think we'll open it up to questions in the last 10 minutes or so.
Thank you so much, Raina and Meghna. That was great. And we do have some questions coming in. And I would encourage folks, if you do have questions, go ahead and type them into the chat window or the Q&A window, either way. And we have a few minutes here to go over them. So here's the first question, which is about natural language processing. So either one of you can answer this one. The question is, are you using natural language processing to automate any parts of your oncologist workflow when you're reviewing and ingesting that unstructured medical record data sets?
Yeah, so thank you. This is Meghna. Thank you for the question. So at Flatiron, we are looking at a lot of machine learning techniques, including natural language processing, to actually identify patients who have maybe a rare mutation. So you can think of we are generally creating these large data sets, these electronic data marts of maybe, let's say, 50,000 lung cancer patients. But let's say we are interested in identifying patients with a particular mutation, and that might be quite rare, such as maybe MSIH or ALK or something like that. And then we use natural language processing to and machine learning techniques to identify patients from that, as a first step to identify patients from those 50,000 large data sets, and then have the abstracting team go in and actually get that information. So it's really a technology enabled abstraction. So we're using technology to extract information about the patients using natural language processing.
Thank you for that. That's great. So next question. This one is directed to Raina. How does your MPH and your background in epidemiology influence your work at Flatiron? And do you recommend a background in data sciences along with public health?
That's a great question. I can honestly say that I probably could not work in this team without the background of my MPH. I think a lot of what you learn in a master's in public health program, and specific really to the epidemiology and biostats training, is really critical on a day-to-day here. So an example of that is when I was in my master's program, I took a class on bias analysis and how that applies to both study design and when you run your analyses, and like evaluating things like selection bias and misclassification bias and all of these other ones really do have a large part in the work that we do here in Flatiron, because every decision that we make about how we're going to abstract or obtain data from the patient record, to answer all of those questions, we inherently have to consider some of these biases. So I would say, yeah, I definitely use my epidemiology skills really on a daily basis.
Great. And the second part of that question was about do you recommend a background in data sciences along with public health?
So I'll speak to myself. I do not have a background in data science at all. Really, what I had learned about data analysis came specifically from my master's program. And even so, in my master's program, I think they taught us analysis methods in coding in SAS, but here at Flatiron, we typically code in R. So even then, I think like your background doesn't have to be specifically and immediately translatable to the work we do on the QS team. There is, you are allowed some time to develop those skills and learn once you first join.
Great, that makes sense. So my next question for either of you or both of you, how did Roche's acquisition of Flatiron impact your work?
Yeah, maybe I can take that. This is Meghna. So Roche's acquisition of Flatiron. I mean, the short answer is that it hasn't impacted our day-to-day work at all, and that's mostly because Roche has really left Flatiron as an independent company to essentially with like little to no meddling around in our business. So on a day-to-day level, it hasn't kind of impacted in the processes and so on. Maybe what it has impacted, it has only strengthened. Roche was always our customer. It has now only strengthened our collaboration with them, and where it has provided Flatiron opportunities where we can leverage Roche's network and expertise in oncology to actually further or improve processes at Flatiron.
Okay. Next question for either of you is, are there any undergraduate internship opportunities at Flatiron?
Right, yeah, so yeah, that's a really great question. And we have two positions currently open, or at least we might be posting very soon for summer internships. So yes, we do have them in the New York office, not in the San Francisco office.
And outside of the quantitative sciences team, they do have undergraduate internship positions for the software engineering team as well.
Okay, and if someone wanted to find those, would they find them at that link you gave us earlier where the positions are posted generally?
That's right.
Okay. Right. So you already touched on using R, but this question is about what programming languages you use to manipulate the data sets. Anything else besides R?
It's really just R.
And then we use a lot of packages like dplyr that allow us to use SQL-like language, but it's mostly R-based.
I see. Okay, we have another question. How could somebody who's interested in a career at Flatiron prepare for the role if they do not have an epidemiology or biostatistics background?
I think, yeah, that's a really great question. So, I mean, I think the main thing that we look at when we try to hire is really, an analytical background. So it doesn't have to be biostatistics. It could be, health outcomes research, or as I mentioned, we have had people who have who are computer science degrees, or we have computational biologists, and so any degree. If you have a degree that is analytical in nature and you have an analytical mindset, I think that's more important than having a specific degree or in a specific discipline. Our hiring process and the steps that we go through kind of are more general. We tend to not ask too many, statistically deep questions, because we understand that, data science and quantitative sciences is a much broader field. So we don't expect our candidates to be absolutely well-versed in every statistical method before coming to Flatiron, but really have that analytical mindset, the curiosity, the innovative thinking. We really emphasize on problem-solving skills. That's more important, the ability to learn quickly on the job, proactivity, communication, collaboration. Those are some of the things that we place much more emphasis on than having a degree from a particular institution or in a particular department.
Thank you. So we have another question. Is there discussion about extending Flatiron's methods to non-cancerous diseases, for example, HIV or dementia?
Yeah, we get asked that question quite often. Currently, I mean, the short answer is currently not. Flatiron as a company is focused on oncology. This is a 6-year-old company. So we really want to make sure that we kind of have a really strong foothold and leadership in oncology. And so currently there are no plans of moving into other indications, but who knows, that might change in the future.
And a follow-up on that one, are you aware of other companies that are using a methodology, anything like yours, to look at other diseases?
There are companies that are doing, similar kind of work using real-world data in ophthalmology and others. So yes, there are, and we are focusing on cancer only, but there are other companies who are doing similar work in other indications.
Great. Well, if someone has quick fingers, they can type in one more question. But otherwise, we're just about done. Let me just see here. Yes, we do have another question. This will be our last one. It says, I saw on the Flatiron page that there's a data insights engineering team. What would you say is the difference between the quantitative science and the data insights engineering team?
Right. Yeah. So there is, so as you know, quantitative sciences, we deal with the analysis of data, statistical, methodologies, and so on. The data insights team really focuses on tool development and looking into building efficient tools, it could be for analyzing the data or for visualizing the data, etc. So, they're not involved in the building of big data sets or statistical methods research, rather creating tools that facilitate some of that research.
Well, thank you, Raina and Meghna. It's really interesting to hear about your work, and thanks to everyone who participated, either just listening or asking questions today. That's all we have time for, but I hope everyone has a really nice rest of your day.
Thank you so much.